Hello, everybody, and oh my god, it's working. It's actually working. My audio is working. Hopefully, I don't know. Please tell me it's working. Someone in the chat. Has a minor miracle occurred? Hello? Someone? Of course. Excellent. Okay, awesome. I'm not muted. This is a miracle. Yes. This is a miracle. Everyone, it is the 2nd of April in Australia, so it's not April Fools. Oh my god. Everyone, write this down. Write this date down. I have finally been able to once not completely screw up the audio. Are you talking about Amanda, are you talking about the chat delay on my chat? Then yeah. I've got it at normal latency, so there's always going to be a bit of a delay on that one. Anyway, by the way, thank you everyone for coming. I do appreciate you all coming to learn a little bit of science. So Ryan Daly, thank you for coming. So basically, Pale Blue Mama, Math Pig, Not Gel Witness, and Time Lord Iro. I hope I'm pronouncing your name correctly, Time Lord. Is it Iro or Iro or something? Can you let me know in the chat? Levi, thank you for coming. Levi, sorry. And Irish Swede, thank you very much for joining in. And I do appreciate everyone for coming. Yeah, Mama, it was because I realized that I was on Top Chat, not Live Chat. And so when I put it onto Top Chat, onto Live Chat, your thing was there. So that's why. And Mofab, did I say hello? So, yeah. Math Pig says normal can suck balls, go ultra low. I really should, but I am terrible like that. Anyway, a couple of updates. If you didn't see on Twitter yesterday, I got bitted by Lilith, which is annoying because it's kind of like, you know, when you scrape your hand against a brick wall and you have like a sore hand, and it's just an irritating kind of thing. It's basically what it is. She doesn't hurt. It's just this irritation more of anything anyway. So, I've got a significant chunk of my finger has been nommed on. So that's an annoying thing. But, thank you, Amanda, for saying that lipstick was bomb on you. Thank you. I haven't put this lipstick on in a while. But it's nice to pull it out. The bright red. I love it too. Anyway, so that's annoying for me. However, it is kind of good because the next time Lilith does a poo, we'll be able to see if she has got any E. coli from me. Hopefully, fingers crossed. I haven't had a chance to go into the lab yet to get her microbes to be sequenced because a week or so ago, there was flooding in Brisbane, so they closed down the university. And I was supposed to go and meet some people to talk about the university. And then they were like shut down because there was a COVID outbreak. Thankfully, it hasn't amounted to anything. It was just a snap shutdown for 3 days. So unfortunately, I couldn't go. So we haven't done that yet, but in the next couple of weeks, I'll start getting results in. So I'm really, really excited about that. So that's pretty cool. And Yeah, not Jar did. That is true. I did pretty much ask it bit though. Like I put my hand near her and she was like, and she's, the thing with Lilith is she's not the brightest spark in the universe. I still love her to pieces. But if you're not actively handling her, if she's not being held by a human, she's in hunting mode. So it doesn't matter if she's outside of the cage, in the cage, whatever. If she's not being handled by a human, she's gonna bite you. And so I put my hand near her and she just went, yoink. So yeah, so that's kind of what happened. She actually went up my finger and then started eating down my finger as well. So she got a good chunk of my microbiome, which is kind of what I wanted. Annoyingly, though, I have to put myself in harm's way. It's no, as I said, it's no different to scraping my hand up against the brick wall. And anyway, so I'm just reading what Amanda Pale Blue Mama said. Angel accidentally got bitten by Leia. He stuck his hands in her mouth, I'm assuming, because he stuck his mouth while she was chewing. I mean, unless he decided to like full on face inhale her, I'm assuming he stuck his hand in her mouth while she was chewing. He's losing his nail and keeps asking me to kiss it better because he's a 17-year-old baby. That is really cute. Anyway, so what was I saying? Yeah, so that's what's happened. I also have to say a quick thank you to Notja. He's actually in my bedroom sort of being really, really supportive because I'm out in the kitchen and so he has to stay in the bedroom for a bit because of his anonymity. So I just want to say thank you and I love you, Notja. You're one of the best people that I have in my life. So thank you. Okay. Oh, yeah, my Fab says the things we do for science. That is true. That is very true. I mean, I have poo in my fridge at the moment. So yeah, so that's a bit of an update, something really, really cool. Fingers crossed in the next couple of weeks I'm going to be able to start sequencing the E. coli and the microbes that are going to come out of her and Thyla as well. I won't get Thyla out because I got her out the other day at this time and she was pretty cranky about it, so I'm not going to do that. Anyway, so that's a bit of an update on my I think. Anyway, so what I have decided to talk about, I had two papers I really wanted to talk to you about. One I thought was absolutely cool. They were talking about using snake venom to as a way to kill antimicrobial resistant Staphylococcus aureus, which is MRSA, or golden staph that most of you might know. And I thought this is freaking cool, but it's a 12-page long study. So it's cool, but I don't think everyone has enough time for a 2.5-hour stream of me geeking out over like AMPS. So I decided not to do that. So what I have decided to do is talk about the genome sequence of avian pathogenic E. coli strain OK01K1H7 shares strong similarities with human extraintestinal pathogenic E. coli genomes. That is not the easiest thing to say. I will be stumbling over myself 100,000 times because, science. Science also is not just difficult in the sense of wrapping your head around things. It is also because it just makes you speak in a language that wants to break your tongue. Okay, so where are we? There we go. That's what I'm doing, hopefully. That's still good. Okay. Blah, blah, blah. Hang on. Let me get back to where I was. Okay. So, basically, E. coli. Remember a couple of weeks ago I was talking about how E. coli strains within there's different E. coli strains within the species. So there's human E. coli, there's avian E. coli, there's crocodile E. coli. If you remember that, and how that we can figure it is just by sequencing and different types of sequencing that was basically the whole gist of that first paper that I read. There is of course E. coli that lives within the intestines and also outside of the intestines, and most of it, as I said, is pretty benign. It lives everywhere. E. coli is easy to grow. It grows everywhere. It's happy and does its thing. But occasionally you can get a pathogenic E. coli. So pathogenic, of course, meaning that it makes you sick, and we can actually see that there's E. coli that lives outside of the gut. So it also depends where it lives as well. And it says here Escherichia, which again I probably am pronouncing incorrectly. Strains that cause the disease outside of the intestines are known as extraintestinal pathogenic E. coli, or XPEC. I'm going to call it XPEC from now on, and include human uropathic E. coli, or UPEC, and avian pathogenic E. coli, or APEC. So most people probably don't know this, but the majority of urinary tract infections are caused by E. coli. And yeah, when I found out that, I was like, oh, that's interesting. Anyway, regardless of the host origin, XPEC strains share many traits. It has been suggested that these commonalities may enable APEC, which is the avian, to cause disease in humans. Here we begin to test the hypothesis that certain APEC, avian, strains possess potential to cause human urinary tract infections through virulence genotyping of 1,000 APEC and UPEC strains. Generation of the first complete genomic sequence of an APEC strain, and comparison of this genome to all available human XPEC genomic sequence. I hope that makes sense. Anybody got any questions? Hello, who's your buddy? And Osmond Plays Games. Thank you for joining up. I do appreciate you. Okay. Where was I? The genomes of APEC-01 and 3 human UPEC strains were found to be remarkably similar, with only 4.5% of APEC-01 genome not found in the other sequence XPEC genomes. Also, use of multi-locus sequence typing, which is what I talked about the first paper that I did, which was, trying to figure out different locuses. Locuses meaning parts of the genome that you sequence. So there's, and where it is in the genome. So think locus meaning like location, if that makes any sense. Use of multi-locus sequence typing showed that some of the sequenced human XPEC strains were more like APEC-01 than human XPEC strains. This work provides evidence that at least some human and avian XPEC strains are highly similar to one another, and supports the possibility that a foodborne link between some APEC and UPEC strains exist. Future studies are necessary to assess the ability of APEC to overcome the hurdles necessary for such a foodborne transmission, and epidemiological studies. I hope I haven't got... Oh, you guys can't see what I'm looking at, can you? No, because, Yeah. There we go. There we go. Why did that cut out? Sorry. Where was I? Yeah, this work... Future studies are necessary to assess the ability of APEC to overcome the hurdles necessary for such foodborne transmission, and epidemiological studies are required to confirm that such a phenomenon actually exists. Again, with epidemiology, it is really important to find out where the source of the disease is. So, yeah. Okay, is MathPig being annoying? If I had a wrench, I'd share an amazing client science that chat that Mama had today, but I'm biased and sleep deprived. Go to bed. Yes, I did just notice a black screen, Notja, because I have to use this just one particular screen just so you can see my face. So yeah, hopefully this isn't going to keep doing what it's doing. Okay. It's giving a bit of background. E. coli is one of the world's most studied organisms and is found at the forefront of advancing technology. Pretty much anything to do with bacterial-based technology is often, not all the time, but is often E. coli. As I said, it's easy to grow. It's very common. We're really familiar with its genomic sequencing. We can clone it. It's pretty good when it comes to technology in figuring those sorts of things out. Where was I? Thus, with more genomic sequences available for E. coli than for any other species, it leads this trend. Thus far, all of the pathogenic E. coli strains sequenced have originated from human hosts, and of course that's led to bias in our knowledge. And because, of course, E. coli can cause illness in other species, such as birds, for example. Consequently, while the genomic analysis of E. coli strains from animals can be justified solely on the basis of E. coli's detrimental impact on animal agriculture, broader justification will also include the potential link between animal source E. coli and human disease. So there's been some links between E. coli and animals and disease. For instance, recent reports of outbreaks of UTI infections have stimulated interest in the potential that E. coli from animals has to cause UTI via the food supply. Since UTIs are one of the most common bacterial infections, of course, I challenge you to find me a woman who hasn't had one, and it makes you quite sick. I have had several in the past, and they're never fun. They're never fun. So of course it's an expensive disease to have. A lot of people get sick. There's time off, and it's not fun. So one hypothesis is that retail poultry harboring avian pathogenic E. coli represents a foodborne source of E. coli clones capable of causing human UTIs. And so they talk about this disease, colibacillosis, which I had to look up last night, and now I suddenly can't remember what it is. And I think it's just an infection that can get the animal really, really sick. I think it's like it's a bit like just a systemic infection of E. coli. Yeah, I just noticed the black screen. So, yeah, that's annoying. Yeah. So apex strains are a subset of extra-intestinal pathogenic E. coli. It is on the animal's skin. It's within its blood system. It's not a nice infection, which is one of the reasons why factory farming is horrendous when it comes to the health of not just the animal but of humans as well. So factory farming, not good at all. Because if an animal is sick, it just spreads throughout the entire thing. And anyway, let's not go on to that rant. Apex strains are a subset of extraintestinal pathogenic E. coli strains, a pathotypic category which also includes UPEC and E. coli causing neonatal meningitis and septicemia. That's not good. You don't want meningitis or septicemia at any point in your life, especially neonatal. as XPEC, APEC, and UPEC both cause extraintestinal disease, share virulence-associated traits and overlapping O serotypes and phylogenetic types. Additionally, there is well-documented history of transfer of E. coli strains and their plasmids from poultry to humans. Okay. So what happens, one of the ways that... see you later, Levi. Yeah, thank you for coming, by the way. Levi says, I'm so sorry to have head out. It is late here, but I'm glad to have finally got to catch you live. Well, thank you very much for coming. I really do appreciate your support. What I was about to say is that one of the things that that microbes evolve, one of the ways microbes evolve is horizontal gene transfer. So basically, one microbe comes up next to another microbe and basically just sticks its like a pilus. Basically, they have sex. So just that has a pilus, it just sticks it in there, shits through some of its plasmid DNA, and then buggers and then leaves. And plasmid DNA contains a lot of sequences such as pathogenicity, and antimicrobial DNA information and things like that. So if you take a colony of E. coli that is not antimicrobial, and then you inject antimicrobial positive E. coli in with and mix it around within that colony, that second colony will eventually get the antimicrobial gene, even though it has never come across or ever had to evolve this gene. So that's really scary, and that can happen amongst different species and even, I think, even different genuses of microbes. So that's kind of scary. Yeah. Yes, Ryan says plasmids are so very useful. Yes, they are very useful. They are basically, a plasmid is like a circular bit of your DNA. So in non-eukaryotic cells, organisms, when a eukaryote is like us, it has membrane-bound organelles. I'm going to try and not speak science these. Okay, so I'm sure everyone has heard of the nucleus of the cell or the Golgi apparatus. They're like organs within the cell. Those organs that are known as organelles, and they can often have some form of membrane or something that's surrounding the organs of this cell, and that's eukaryotic. Prokaryotic is cells that don't have they have like a nucleus of the cell nucleus DNA, which is kind of like a big mush of DNA in the middle of the cell, and then you have plasmid DNA, which is like a circle just swimming around outside of the nucleus of the microbe. And that plasmid DNA is what we look for and what we can play with in regards to a lot of our genetic engineering and genetically modifying it. So that, I hope that helps. I hope I'm making it a little bit, yeah, Ryan Daly makes perfect. Floaty circular DNA, that is literally it. It's, once you start learning about the biology of even just cells themselves, like even really simple cells, it's fascinating. I mean, like, they have their own kind of, what's he calls it? How do I say it? Immune system. They have their own immune system. Anyway, I won't go into that. I'm going off topic. And hello, Stuttering Chris. How are you going, mate? Okay, so, where was I? A recent report has shown that APEC plasmids can contribute to the urovirulence of E. coli from mammalian hosts. So, avian pathogenic E. coli contribute to the urovirulence of E. coli from mammalian hosts, i.e. causes a UTI. These observations have led to further speculation that the APEC might be the source of a UPEC, which gains access to the human colon following ingestion of contaminated poultry. Here we begin to test the hypothesis that some APEC strains are a source of human UPEC by generating the first APEC genome sequence and comparing it to all of the human UPEC genomes currently available. This newly sequenced XPEC strain was chosen for study because of its serotype, phylogenetic group, and virulence genotype were representative of 1,000 well-characterized APEC and human UPEC isolates. Darth Calculus says, I know so little about prokaryotes. The life science book always goes straight to eukaryotes and organelles. Yeah, it's I kind of understand, because a lot of people really don't care too much about microbes because, I want to learn about my species, which, yeah, I understand that. But prokaryotes, honestly, I keep saying, microbes are going to save the world. Seriously. Or they're going to destroy it, as we... Okay, I think I'm back. Sorry about that. Okay. I'm back. Awesome. Okay. Sorry about that. OBS just crashed. Okay, what was I saying? Yeah, so I was saying that microbes will be the end of us and then the computer crash. So I think there's probably a little bit of jealousy towards microbes from my computer. All right, I'm going to try and get through this reasonably quickly. So they chose APEC-01 for sequencing because it was based on possession of traits, characteristics causing extraintestinal disease in poultry and in humans. APEC isolates were obtained from lesions of chickens and turkeys clinically diagnosed with colibaciliosis from various locations within the US. And the UPEC strains were isolated from cases of human UTI. They were kindly provided by Paul Carson, so hopefully Paul Carson didn't actually have the UTI. Hopefully he just was a doctor that had people with UTIs, because he was at Meritcare Hospital, Fargo. Or they were from the VA Medical Center, Minneapolis and Minnesota, I think. MN? I don't know my the shorthands for things. Please tell me I'm still... Yes, I am. Excellent. A subset of both groups of isolates have been described previously for embryo lethality studies. 3 additional APEC isolates were used. APEC V1, O non-typable, and APEC V2 and V7, both O2. So that was going back to what I was talking about my first stream on this. For one-day-old chick studies, 3 strains previously characterized of being high, intermediate, and low lethality towards birds were used. All isolates were serogrouped through the E. coli reference center, and were screened for traits associated with APEC and UPEC virulence using the techniques described below. Organisms were stored at negative 80 degrees Celsius in brain heart infusion broth. With 10% volume-to-volume glycerol until use. Test and control organisms were examined with the presence of a variety of genes known for their association with APEC or UPEC virulence by using multiplex PCR procedures which have been previously described. Hello, Passionate Kuma? And... Okay. So the genomic analysis was used as 2 random shotgun libraries of APEC-01 were created. So basically what shotgun libraries is that they've taken a sequence of DNA and they've just basically broken it up into smaller sequences, and then going by how they overlapped, they were able to figure out where they attach to each other. So you might have a particular sequence, multiples of particular sequence, and you just break it up into random places. So you might break it up, let's say my fingers were the breakup. Some might be these three breakup, these three have broken up in one sequence, and then they might split in half, and then the other ones, these three broken up. And so when they sequence the genome, they can tell that these two and these two fit here because of these three are the same, and then these three are the same. So that's kind of what it does. They sort of splatter it and then stick it back together, and that's how they sequenced it. Okay, shotgun sequencing was performed on small insert library by MWG Biotech using AB13. Basically, I won't go into that. And the FOSMID library was also end-sequenced. Finishing was accomplished using the pooled primer technique described by Tetlin et al. And used elsewhere. The final gaps were closed using standard PCR. So they basically did multiple sequencing capabilities. Sequencing reads were assembled using SecMan software from DNA Star. Within the plasmid sequence were identified using gene, blah, blah, blah. I'm going to just skip them over most of those. Okay. ORFs corresponding to proteins larger than 66 amino acids in size were identified. Translated ORFs were then compared to known protein sequence by using BLAST. ORF, I can't remember what that stands for. If somebody could let me know what an ORF is, because I'm afraid to look anything up on the internet right now. Okay. Those with greater than 90% amino acid sequence identity over greater than 90% of the sequence were considered matches. So they allowed a 10% variation. The DNA sequence was also analyzed to identify non-coding features. The G plus C content of original ORFs was analyzed using emboss, insertion sequences, and repetitive elements. So yeah, that's basically how they figured that out. Oop, going the wrong way. Open reading frame. Thank you, Ryan Daly. Open reading frame. Okay. Okay, so going back to that. Don't crash. Properties. On. Okay. Okay. Okay, so what are we talking about? So basically, from what I'm seeing here, I'm trying to just skip over the really speaky ones. The really speaky ones. I'm skipping over the overly repetitive bits. So multi-locus sequence typing was used to determine the genetic relatedness of the fully sequenced E. coli genes. And they had 7 housekeeping genes were extracted from sequence database for all these genomic sequences, concatenated to produce a contiguous sequence of 9,115 base pairs and aligned by the neighbor joining method with 1,000 bootstraps iteration using cluster W. I like reading these, I know what it means, but I can still understand why a lot of people just don't get science, because a lot of these words just are completely random. Even I, like with my training and everything, I did have to take a whole year off last year because of COVID, and there's a lot of these things where I'm like, oh geez, I'm dragging these things out of my head. So yeah, I can kind of understand why people think that scientists are a hoity-toity, And everyone's like, oh, what would the experts know? Because they don't know what they're talking about. They don't understand what the experts are talking about, and therefore the experts are wrong. Okay, so APEC-01 and several other well-characterized. So for people who've just joined, APEC is avian pathogenic E. coli, and UPEC is uropathological E. coli. So APEC-01, several other well-characterized APEC strains were assessed for lethality in chicken embryos by inoculation of 500 colony-forming units. So basically that's just a colony-forming unit is a microbe that forms a colony. So of overnight washed bacterial cultures into the allantoic cavities of a 12-day-old embryonated specific pathogen-free eggs. Phosphate buffered saline inoculated and uninoculated embryos were used to control. Embryo deaths were recorded for 4 days, so that's a bit sad. But this is what we unfortunately have to do for science, and of course the ethics department will have their words. Okay, the ability of APEC-01 to cause avian colibacillosis, which is an infection caused by E. coli, was confirmed as chicks, as described previously. Day-old chicks vaccinated only against Merrick's disease were obtained from a commercial source and were divided into groups of 10 chickens each in Horsfall units at the Livestock and Infectious Disease Facility at Iowa State University. Chicks were provided food and water ad libitum, so whenever they needed it. Chicks were inoculated intracellularly with 0.1 mils of bacteria suspension containing 10 to the 7 colony-forming units per mil of APEC-01 with approximately 10 to the 7. So that's 10 with 1 with 7 zeros after it. Phosphate buffered saline subcutaneously in the back of the neck. Chicks were monitored for 7 days. Deaths were recorded, and the survivors were euthanized and examined for macroscopic lesions. So they were just trying to figure out how deadly this was for these birds. Organisms were classified as being low, intermediate, high virulence based on the mortality and gross lesions related to colibacillosis. 3 APEC strains representing each of the pathogenicity groups, as kindly supplied by Sandra Cloud, University of Delaware, were used as controls. And let's hope it doesn't crash again. Okay. And so the biostats, a cluster of analysis of the genotyping results of the isolates tested was performed using the average linkage method. Blah, blah, blah. And they're just talking about, they're just talking about the patterns among the isolates and trying to figure out what's what. Okay, so, and they completed the sequence of annotation of APEC-01 has been deposited into the GenBank under a session number and that number. So basically, they've sequenced it. And they've put it under, they've just basically put it into a database from basically, could be wrong, but I hope not. Ryan Daly says, you biology people love those abbreviations. Can you imagine saying open reading frame 400 times? There's a reason for it. So basically says they should make a dictionary of scientific words for common folk so we can understand them better. That would be good because that would be useful for non-scientific people, for scientific people as well. Everyone would enjoy that. I'm sure they've got something like that somewhere. Anyway, so talking about choice of strain. Extensive virulence genotyping was performed on large populations of APEC and UPEC strains in order to identify an APEC strain for sequencing. This is done in an effort to identify an APEC strain that was classified in a serogroup, phylogenetic group, and virulence genotype characteristic of both APEC and UPEC. Serogrouping, phylogenetic typing, and virulence typing is for nearly 40 APEC and XPEC associated genes were performed in Fuck. 500 APEC and 500 UPEC isolate. So basically they just got 500 avian pathogenic E. coli and 500 euro whatever they said the word was, pathogenic E. coli. They isolated those and they looked at the at certain parts of the DNA of these bacteria. Oh, I'm seeing no. Why am I seeing no? Am I still streaming? Should be. Okay. Oh dear. Okay. Some of the genes examined include those encoding plasmid-associated virulence factors. Remember how I said plasmid was different to nucleoid DNA? So they're both types of DNA. Just plasmids are, like, as I said, a circle of DNA within the microbe. And so the genes examined included encoding plasmid-associated virulence factors such as the increased serum survival gene, the aerobactin and salmonellin siderophore system genes. Don't ask me what that means. Aerobactin, I would assume, is the gene that allows it to be an aerobic and space aerobic bacteria. So you've got anaerobic and aerobic bacteria. And aerobic bacteria are bacteria that can survive in the oxygen, and anaerobic gets killed by oxygen, which is why they think one of the reasons why they think life started right deep down the bottom of the ocean is because of anaerobic bacteria. Okay, where are we? But salmonellin, I'm not entirely sure what that is. I'm just having a look. Okay, thank you, Ian, for letting me know that I'm still streaming. Okay, where were we? Chromosomal virulence associated genes were also sought, such as the vacuolating and autotransporter gene, or VAT, and the PAP, operon, and the Yersinia bactin siderophore system genes. Yersinia bactis is it is also a why pest? Yeah, Yersinia bactis is the Black Plague is Yersinia bacter pestis. So Yersinia bactin siderophore system genes, I would assume might have something to do with pathogenicity. Oh god, it's hard to speak, isn't it? And the IRA or iron responsive element gene. So iron is used a lot in biology for reasons. A cluster analysis of these genotyping results separated the APEC and UPEC isolates into two primary clusters and several subclusters. One of the two major clusters contained primarily APEC strains, characterized by the position of plasmid-associated virulence genes. The second major cluster contained primarily UPEC strains, characterized by virulence genes, which have been localized to chromosome-encoded pathogenicity islands. Any questions? Foppish Dilettante says anaerobic. Yeah, it's it's hard to say anaerobic when you mean aerobic. Yeah, it's okay. All right, so I'm gonna skip through this a little bit because a lot of it is... I'd love to show you what I'm seeing. Okay. And properties. There we go. Don't know why it keeps making me do that. This is interesting. It says APEC-01 was selected because it contained traits typical of both APEC and UPEC. APEC-01 is a O1K1H7 strain that was originally isolated from the lung of a chicken clinically diagnosed with cholesepticemia. The O1 serogroup is one of the more commonly occurring serogroups among both APEC and UPEC strains. O1 strains are also known to cause other important conditions in animals and humans, and a recent report of inter-household spread of UPEC strain involving family members and their dogs suggests that such strains may have zoonotic potential. Despite the widespread contributions of O1 strains to human and animal disease, no genomic sequence of an E. coli O1 strain has been described prior to this report. Foppish Dilettante says, Yersinia would make a good name for my next chicken. Yersinia is a really cool word, actually. I like that word. Yeah, it's a cool word. A lot of people like the word chlamydia as well, if you're not a native English speaker. Chlamydia and diarrhea are also really commonly considered to be pretty words if you're a non-English speaker. And then you tell them what they are and they're like, ew. APEC-01 is also classified in the B2 phylogenetic group, which is closely associated with extraintestinal virulence in E. coli. Prior to sequencing, the ability of APEC-01 to cause disease in birds was confirmed by intratracheal inoculation of one-day-old broiler chicks. According to the scheme used by Rosenberger et al., using this model, APEC-01 was categorized as being highly virulent for chickens, and that is table 1. And you're not going to see what... Oh. And going back. Awesome. These plasmid genome thingies are quite pretty. I've always thought they were... So the overview of the APEC-01 genome was associated and finished at approximately 8-fold coverage. It contained a 5,082,025 base pair chromosome, and unlike other sequence XPEC strains, contained 4 plasmid, totaling 565,500 base pairs in size, giving it a total genome size of 5,647,625 base pairs. That's a lot. The presence of the... It's a plasmid! It is a plasmid. I love plasmids. They're really, really cool. The presence of these plasmids was not surprising, considering that plasmids are a defining trait of APEC pathotype. Such plasmids have also been identified among UPEC strains, although a lesser extent. 2 of APEC's O1 plasmids have been previously described. A 174,231 base pair IncFib colicin encoding virulence plasmid and a 241,348 base pair resistance plasmid encoding resistance to 8 antimicrobial agents. The remaining 2 plasmids, PAPEC-01 CRIP1 and PAPEC-01 CRIP2, are 101 and 201 kilo base pairs in size and kilobytes, respectively. These plasmids share limited identity with Yersinia type plasmids, code mostly hypothetical proteins, and do not confer any apparent phenotypes. Cool. So I thought that was kind of cool, the fact that E. coli actually has Yersinia style or Yersinia type plasmids, which is, remember when I said plasmids can be transferred between bacteria even if they're not of the same species? That's pretty cool. Mofab says, why do some of the nastiest diseases have such beautiful names? I know. What was another one that I thought was really pretty? I'll have to think about it. Ebola is actually a pretty name as well, if you think about it. It's very pretty. Pretty names, but they kill people. Going back. Okay, so the G plus G content of the APEC01 chromosome is found to be 50.5%, which is comparable to that of other E. coli strains. However, the plasmids of APEC01 had a variable G plus C, so that's glycosine and cytosine content. So that's, of course, in the DNA, ranging from 44.6 to 49.6. Within the chromosome, regional difference in G plus C content could be discerned, with the differences occurring primarily within the genomic islands, which are defined here as regions of APEC01 greater than 4,000 base pairs and that are not printed in E. coli K12MG1655, and codon adaptation indices reflect such differences in C plus G contact, with differential codon usage also occurring within genomic islands. Okay, so codons, if you don't know what they are, they're 3 base pairs, and that 3 base pairs basically get turned into an amino acid when the DNA gets transcribed from DNA to mRNA to RNA. Yeah. So that's what a codon is. A codon is 3 base pairs that have been transcribed into an amino acid. I really feel like I'm not explaining this very well. And if you don't know what an amino acid is basically the building blocks of a protein. So if you're going to say a protein is like a house, the amino acid is the brick, and the codons or the base pairs are like the sand in the brick, if that helps. All right. I think someone's heading off. I can't see. Oh, no, somebody's not. Okay. All right. And what they found is that APEC01 is highly similar to human XPEC, which is like extraintestinal pathogenic E. coli. At the time of this publication, the genomic sequences of the 3 human XPEC strains were available for genomic comparative genomes. So one strain was isolated with the patient with pyelonephritis, which I've got a feeling is an infection of the kidneys. Another strain was isolated from a patient with a complicated UTI, urinary tract infections. Another one was isolated from a patient with an uncomplicated cystitis, which is, again, a type of UTI. And comparison of these E. coli sequences was quite revealing, which is table 3. Hang on, I'll just roll down, and there we go. That's it. That's kind of telling you content of APEC01 open reading frames, so APEC percentage, so 100 is, of course, APEC01, but they're all in like 93, 90, 87, 86, and 78%. So that APEC01 is that what they were using to identify the sequence of the microbes that was causing disease. Okay, now where is it? Within the APEC01 chromosome, 4,467 ORFs were identified. 78.3 were of which part of a backbone common among all sequence E. coli strains. Yeah, so that's pretty cool. That's really interesting. They were finding that the avian pathogenic E. coli is very similar to the extraintestinal pathogenic human E. coli. Yeah. So what I think from what they were saying is that eating chicken that was infected with APEC caused genome swapping through the plasmids, which then infected the humans, which may have caused pathogenic E. coli to occur from APEC. Yeah, so as it says here, such a backbone of genes might be essential in the ability of XPEC to cause an extraintestinal disease or to survive the extraintestinal environment, as genes within this XPEC backbone include the loci thought to play a role in XPEC virulence and /or fitness. Some of the regions identified within this backbone include the Acinetobacter and PAP operons and other places. I'm just going to skip that because that's just a bunch of gibberish for 99.8% of the world's population. In addition to the characterized islands within the common XPEC backbone, 16 other islands were identified and were simply termed XPEC islands, encoding most hypothetical proteins. Future studies will be needed to ascertain their functions and roles in virulence and persistence, if any. All right. So those are all the tables. Okay. So they're talking about genomic islands, okay. Here's some interesting parts when they're talking about phage-related stuff. So all of the APEC-specific islands are either composed of prophage-like elements or are found adjacent to prophage-like elements. These phage-related APEC-01 specific regions will require further study to determine if they might be used as markers to distinguish APEC from human XPECs. So phages, if you don't know what they are, they're like viruses that infect microbes. And these phages may actually end up helping us with destroying, with overturning the, antimicrobial resistance. Because what happens is when a microbe evolves, they to cause antimicrobial resistance, they lose a lot of their ability to kill off the phages. So if you can find phages, which is this virus that injects in the microbe, it has to try and do a balancing act. So you've got the phage that comes in, kills the microbes. So the microbes have to evolve to get rid of the phage, and by doing that, they lose their antimicrobial ability to resist those. So people with severe illnesses caused by by microbes may have phage-based antimicrobial disease. So basically, they just inject the person with the phage. The phage is very specific to a particular type of microbe. The phage kills off the microbe, and then they give antibiotics to kill off the ones that survive the phage, but won't longer survive the antibiotics, which is pretty cool. Is that plasmid absorption an epigenetic change? It's a horizontal genetic change. So it's not, I don't think you could really consider it to be epigenetic. I'm not entirely sure, but it's a horizontal gene transfer. So basically they're just putting some genes across. It'd be like you touching somebody, and if you've got red hair and you touch somebody and then the next 6 months later they have red hair, it's the same sort of thing, even though that's not possible. Okay, so we're almost there. As I said, there is another study that I wanted to read to you, but as I said, it's 12 pages long and it's huge, but it's cool. There have been previous reports of virulence-associated genes or operons occurring at specific locations in the APEC genome. Comparison of these regions genomic islands of APEC-01 and other sequence XPEC strains helps to further define these groups. For instance, Limbopolis et al. Recently described the STG fimbrial operon occurring in APEC-078 strain. This operon contributed to the ability of that strain to colonize the respiratory tract and was present in 45% of the APEC strains examined. Also, this operon was inserted between the GMLMS and PSTS genes in APEX that number. However, this operon was absent in APEC-01 and other sequence XPEC strains, and no other insertions were found within this region. Similarly, Chowika, I butchered that name, and all described a virulence-associated genomic island at the cell C locus in the APEC-02 strain. In APEC-01, the 3 other sequence XPEC strains, although a different island is inserted at this location, whereas the island described by Chioka, ruined that name, is absent. So based upon virulence genotyping, APEC-01 had one of the highest counts of virulence genes of all of the XPEC strains we examined. Despite its complement of virulence genes, it was significantly less virulent than an embryo assay of virulence other than APEC isolates, which contained fewer XPEC-associated virulence genes. Therefore, it would be seen that the number of PAIs and/or virulence genes in an XPEC strain is in itself insufficient to predict virulence, at least in this model. Instead, these results may be related to some minimal XPEC backbone that is required for virulence. Okay. Ryan Daly says, isn't EPI change the result of promotion or demotion of specific gene expressions? Plasmids are not quite EPI, so epigenetic change. Ryan, I should probably have you on. You can just... You're doing great. Okay. They're just saying it's clear that the genomic basis of XPEC virulence remains incompletely understood. We're almost there. Okay, I'll just skip through some more because, I'll just skip through some more. APEC-01 and other sequence XPEC are phylogenetically related. Phylogenetic, of course, is the tree of life. The phylogenetic tree. Protein blast searches of the ORFs of APEC-01 indicated that it is very closely related to other XPEC strains. Ryan, you're not boring. Physicists are very interesting. I've got a good friend, almost a doctor. I'll shout him out if you want to go and watch a good Australian physics channel. Look up Almost a Doctor. He's a good friend of mine. Okay. So I don't think we really need to know this. This here is the phylogenetic tree. That's the phylogenetic tree there. All right, we're up to the conclusions, and we're also up to. Okay. So the conclusions. Overall, this study provides compelling evidence that the genomes of APEC-01 and several sequence human XPEC strains are very similar, thereby providing suggestive support of the hypothesis is that at least some APEC, avian pathogenic E. coli strains, are a source of UPEC, urinary pathogenic E. coli strains causing human UTI. However, these findings must be interpreted within their context. That is, APEC-01 was selected because it belonged to a serogroup and a phylogenetic group implicated in both human and avian disease, and it belonged to a virulence genotyping cluster containing a mixture of APEC and UPEC strains that were typified by a possession of several common XPEC virulence factors. This work makes no claim at all, or even most, human extraintestinal infections are derived from poultry source E. coli. Rather, this work provides evidence that at least some human and avian XPEC strains are highly similar to one another, and it supports the possibility that a foodborne link exists between some APEC and UPEC strains. However, validating this hypothesis requires an assessment of the ability of APEC to survive and persist on retail poultry, to traverse the human intestinal tract, and to ascend and colonize the urinary tract. Furthermore, epidemiological studies will be required to confirm that such a phenomenon actually occurs. While APEC-01 was highly similar to other sequenced human XBEC strains, it was not identical to them. As observed with all E. coli strains sequenced thus far, APEC-01 contained a percentage of unique genomic regions not occurring in the other sequenced E. coli genes. Before definitive conclusions can be drawn about the validity of the hypothesis that some APEC strains are involved in zoonotic disease, the distribution and function of these APEC-01 specific regions should be addressed. It is possible that these regions limit the disease-causing ability of APEC-01 and perhaps other APEC strains to avian hosts. However, the sequencing data available at this point imply that all E. coli strains possess at least some unique regions, regardless of their source. A case in point is the 4 UPEC genomes which have been sequenced. While the chromosomes of these strains were quite heterogeneous, they were isolated from patients with similar types of disease. Thus, a better understanding of XPEC genomes and a more lucid definition of the XPEC subpathotypes are necessary in the future. In conclusion, this study provides the first genomic sequence of an APEC isolate and of any animal source E. coli isolate. Based on comparative genomic analysis, it appears that APEC-01 shares extensive similarities with the human XPEC, and study of its sequence has resulted in a refined understanding of the E. coli pangenome and the XPEC backbone. These extensive comparisons of APEC and UPEC leave open the possibility that some APEC strains represent a potential foodborne source of human UPEC. Regardless, this study provides an APEC genomic sequence on which further testing of this hypothesis can be effectively based. Thus, the sequence of APEC-01 is another stepping stone towards controlling XPEC-caused disease in avian and human hosts. So even though the genomes are pretty similar and there could be a connection, they're not saying that there is. That's all. All right, so that's the end of this paper, because of course you've got the acknowledgements, and unless you want me to just read so you can fall asleep, I can, but I don't like the sound of my own voice. Yeah, so if you've got any questions about that, let me know in the chat. In the meantime, as if you missed the beginning of this stream, I was just showing off my war wounds from Lilith, who decided to give me a nom. I hopefully should be in the lab next week or the week after, finally starting getting some of these results, which I'm excited about. Ian Burns says, so it's likely that I'm not a chicken and still a ferret. Yeah, pretty much. I would agree with you on that one. It's 10 o'clock. That sounds great, actually. From the High Apostate. Hello for coming, by the way. And, oh, don't forget to like the video, the stream, by the way. If you haven't done that, just give it the thumbs up. Also, with my other channel, Navadia, I am coming back. I've got a feeling that because of all the YouTube stuff that's going on, reading a paper takes a lot of brain power. So I might end up just doing a fortnightly video. This one I'm working on right now is how the media and we as a society reacts to people who fight back against people in power. I'm going to be talking a lot about Meghan Markle. I know it's a controversial thing, but also people such as like Bill Clinton, not Bill Clinton, Monica Lewinsky, Dr. Fauci, and people who had the audacity to talk out against powerful people and how unfair it is that they get a lot of hate. Anyway, so if anybody has any questions, I'm just going to continue rambling to myself. I currently have a troll on my other channel as well, telling me I'm old and I'm losing my hair and I need plastic surgery. I'm surprised they haven't started telling me I'm fat, but we'll give it a bit. Yeah, they were like, thought that was great because, you know, misogyny isn't a thing. and yeah, anyway, so I'm going to sign off. I hope you guys enjoyed that. I thought it was pretty interesting. I really wish I could have done the other one, that 12-page long one. I might actually make that a scripted video to talk about that in my other channel because it's so cool. Yeah, Ryan, what the fuck was basically it? Yeah, I mean, if you go on to my last 3 videos, including the short that I put out, and just sort by newest comment first. Yeah, who's your body says that Incel just had micropeen. Apparently, Faye the Most Gracious has that same troll saying the same shit about her. I would probably put money on them not doing that to a male, to be honest with you. No, I'm very tempted to ban them, actually. Ryan, I didn't realize that they were trolling, actually, when I interacted with them, and then because they said that my hair is getting thin and I'll need to have a wig. And that was on my anemia video, and I've completely forgot that anemia can actually cause your hair to fall out. So I was like, oh yeah, that's a good point. And then I saw, like, immediately after that, they were saying some pretty shitty things to me, and I'm like, fuck. Yeah. Oh, Ryan, no, I'm not old. I mean, this, none of that's actually, it hasn't upset me. It's just pathetic people. Anyway, I'm going to go, and I hope everyone, Ryan, possibly a funding, I would just say it's just a troll, but anyway, I'm going to go, as I said. I hope everybody has a lovely day, evening, night, whatever, and don't forget scandium, iodine, nitrogen, and cerium.